Good afternoon, everybody, and thanks for still being here at the very last presentation of today. Thanks also to the organization for inviting me to tell something about the relatively small pilot study that we did with be-TSC in TSC. Yeah, my name is Dorinde van Andel. I'm a psychologist working at the UMC Utrecht, so the University Medical Center in the Netherlands, at the Department of Psychiatry. And I'm conducting research within the sensory processing program, where we look at different or aberrant sensory processing over different neurodevelopmental disorders, such as ASD, ADHD, epilepsy, and also TSC. And today I would like to show you the results of the Batch study, which we just finished. I'm just starting to analyze everything and write them up, but yeah, I'm going to show them anyways today. Yeah, so to start, why treating behavioral symptoms in TSC? I think that point has already been made fairly clear today. But if you look at the current medication studies, there's a lot of emphasis on tuber reduction and also on seizure management, which makes sense, of course, as approximately 80% of patients with TSC experiences seizures and epilepsy. But the TAND symptoms, which are mentioned already, so I won't go into them, are really a huge burden as well. They have a significant impact on daily life functioning and on quality of life. However, at this moment there are no cognitive or behavioral treatments available. And that's why we started the Batch study, because there was evidence pointing towards the efficacy of be-TSC in TSC. Yeah, what is be-TSC? It's a water pill, a plus pill, we call it in the Netherlands. It removes extra water from the system, but it also lowers blood pressure. But in the brain, it has a kind of a different function. It's an NKCC1 antagonist, so it's a chloride importer antagonist. And that means that it has influence on the intracellular chloride levels. It can lower these levels. And as you probably all know, GABAergic transmission depends on intracellular chloride levels, showing a progressive shift from depolarization to hyperpolarization during cortical development. So kind of from excitatory transmission to inhibitory transmission. But in some pathological states, these high chloride levels remain. And that was also indicated in TSC, as there were elevated NKCC1, KCC2 ratios found, and both in animal models, but also in human tuber and perituberal tissue, which was suggestive of be-TSC's efficacy. So to kind of carry on with this hypothesis, we wanted to test whether be-TSC would have a good influence on behavior in TSC, and also whether this would be reflected in EEG measurements. So that's why we started the Batch study, an open-label pilot study. We actually first aimed at including 20 patients. We finally included 15 patients between the ages of 8 and 21 years old. We did a variety of different measures. So we used EEG measures, different paradigms. I'll tell you a bit about that later. Neurocognitive assessment. We did a lot of different behavioral questionnaires. So like the ABC, so the Aberrant Behavioral Checklist, but also more autism questionnaires, the SRS. And of course, also the TAND checklist. And we looked at epilepsy-related variables. Yeah, we tested the patients at baseline, treated them with be-TSC for 3 months, and afterwards we had a washout phase again. And then we measured them again. So how did it look like for the patients to participate in the study? Well, first they had a baseline visit. So we had an anamnestic interview, sensory profile interview, IQ test, and physical examination. Then if their intellectual capabilities were above certain level, we did neuropsychological assessment and EEG assessment. This wasn't possible for all the patients. Some patients just kind of like, ripped off the EEG electrodes. Then we combined all the measures that we gathered and gave the parents and the patients kind of a personalized sensory profile. And that's when we also started with the bumetanide phase, so the study phase. We give them 2 tablets per day with potassium chloride suppletion, and in total that 5 times they were coming to either the UMC or to their GP for blood samples and physical examination. Then after 3 months, the medication phase stopped and they came back to the UMC and we repeated the measurements. And then after a month, we did the same. And then afterwards, we combined all the measures again, so from the EEG, neuropsychological, and the behavioral measures, and we gave this back to the parents again and looked what they wanted at that moment. So a little bit about our cohort. As you can see, age ranged from 8 till 21 years old, and IQ from below 40 to 107, so that's quite a big spread. Then most of the patients had focal epilepsy, and most of them were taking an anti-epileptic drug. And you can see also that there were a lot of psychiatric problems in our cohort. And 2 of the patients we didn't include in our analysis. The first one was last to follow up, did complete the whole medication trial, but didn't come back for the other measurements. And one of them actually dropped out at day 50 due to aggressive behavior. And this was actually the only patient that dropped out during the trial. So I think in general the drug was quite well tolerated. Then the results. So for our primary endpoint, which was the Aberrant Behavioral Checklist Irritability subscale, the ABC-I, we saw a significant improvement on average. And you can see as well on the individual graphs that all but 1 patient showed improved scores. On the ABC hyperactivity subscale was the same, so irritable and hyperactive behavior seemed to improve with bumetanide, and there was no significant change after the washout period, although you can see that there's a tendency towards normalization. Then social behavior we measured with the social responsiveness scale. We saw a similar effect, so a significant effect of after the 3 months bumetanide, which was also reflected in the individual graphs, and for repetitive behavior it was the same. But you notice that the spread is quite broad, so we saw a lot of different patients, and that was also reflected in the individual graphs. Then the effect on sensory processing was actually not that clear. We didn't find significant improvements on the different sections of the sensory profile. We did see a tendency towards improvements, but they were not significant. And on the brief, the behavioral rating inventory of executive functioning, it was only filled out by 9 parents. We did see an improvement of the total scale, but it's such a broad scale and it measured so many different things that I wouldn't interpret this as a significant outcome. Then we did some EEG assessments. So we used different EEG paradigms to look at automatic brain responses, which is a non-invasive way. We used 64 electrodes for that. And we used 2 different kinds of EEG. So we used event-related potential, so ERPs. So that's looking at the EEG signal after giving an auditory stimuli, and we give that auditory stimuli repeatedly in the paradigm, and then kind of the small fragment of multiple trials, the mean of that's your ERP. And then we also looked at resting state EEG, so EEG at rest, when the participant was with their eyes closed or with their eyes open. I'll show you 2 paradigms. The mismatch negativity paradigm, which we measured with an auditory oddball paradigm. And this is kind of a measure of automatic sensory discrimination, or automatic orienting reflex, they call it in the literature. So what the patients hear is they hear kind of a continuous stream of sounds, of standard sounds of 1000 hertz and 50 milliseconds. And this is interspersed with sounds that are deviant. So they're either higher, so a frequency deviant, or they're longer in duration, so a duration deviant, or a combination of both. And what you normally see is that, we calculate the difference wave, so the difference wave between your response towards the standard sounds and your response towards the deviant sounds. And normally you see that your response towards the deviant sound is higher in amplitude. And a greater amplitude of this difference wave is seen as a better discrimination. It's a proxy for better discrimination. So when we look at our sample, we couldn't statistically test this because the cohort was fortunately too small, but actually we don't see clear changes. We see on an individual level that the measures, we see individual changes on the EEG, but we could not interpret them, and they also don't look like they normalize. So no changes for the mismatch negativity. Then we looked at P50, which is a measure of sensory gating, so sensory filtering, and we use a P50 suppression task for that, which is done in a paired click design. So the participant constantly hears 2 clicks, and really in close proximity of each other. And normally the response to the second click, which is kind of the irrelevant click, is inhibited. And that's what you see here as well. So you see a smaller amplitude towards the second click. But sometimes you don't see that. And you see that, for example, also in patients with schizophrenia. You see that a lot in the literature, that the brain response towards the first click is similar than towards the second click. And in our cohort, we actually saw on average a tendency towards normalization, because a reduced ratio is improved sensory gating. However, if you look at the individual graphs, it's so spread, and we couldn't reliably analyze 2 data points because of the EEG. We need to have more participants to really say whether this is normalization after bumetanide. Then lastly, I would like to show you some data from a novel EI marker, which will be published hopefully in 3 weeks. And this is assessing neuronal information processing at rest, and it's derived from criticality dynamics of cortical oscillations. And it's been validated both in our lab and in other labs in clinical and in non-clinical samples. And here I would like to show you that it's sensitive to treatment effect. You see that most, like 6 out of 9 patients show an effect after bumetanide, and the values also go towards the value 1, which is a proxy of balanced excitation inhibition. And this effect was actually lost again after the washout period. Then quickly, other variables that we looked at, it didn't seem to have an effect on epilepsy. So the epilepsy frequency of the seizures remained stable. Adverse events, there weren't a lot of adverse events reported. The only ones that were reported were expected and reversible, so they could be resolved quickly. And also really important, we did the TAN checklist during every measurement, and the parents and patients both were predominantly positive about the bumetanide use. And at this moment, 8 of the 14 patients are continuing to use bumetanide off-label. So to conclude, and I would first like to say that this is an open-label pilot study. So it's not an RCT. We weren't blinded, so I think it's really good also to keep that in mind when looking at the results. But for the patients that we tested with a broad IQ range and age range, bumetanide seemed to improve the social, repetitive, and irritable hyperactive behavior. And it looked like there was a tendency towards deterioration after the washout, but this was not significant. We didn't see an effect of bumetanide on all the EEG measures, but probably the most sensitive measure is the EI marker. It didn't have an effect on seizure frequency, and probably most important, the parents and patients were really positive about being in the trial. I think with that, I would like to stop because it's already pretty late. Thank you so much for your attention, and I'll be happy to take any questions.
Thank you very much, Dorinde, to give us this detailed feedback on your work. Is there at the end of the long day? Is there still a question for Dorinde?
Everybody wants to go home.
Everybody. Okay, there's Anne. Maybe.
I was just wondering, listening to Petrus earlier today when he briefly mentioned the findings of the Throne trial, that's mTOR, of course, but since there was that was placebo-controlled, and so in the placebo group there was a 40% improvement on the neuropsychological evaluations, probably due to retest or learning effect and being more familiar with the environment of the clinical trial. I just wonder to what extent you think that might also be at play when interpreting your results, or do you think that because of the setting, this is something that was less present?
Yeah.
I just wonder what your thoughts were on that.
Yeah, no, really good comment. When you talked about that study, I was immediately thinking the same thing. So I think it's really good to, when we're analyzing these findings, to interpret it and also put it against like historic findings of placebo-controlled trials, to look what their improvement was on. For example, you have the data from similar like behavioral questionnaires, and if we look at the placebo effects of those to put it against it, and then look if it's still significantly higher. So we're definitely going to do that. I do think it's maybe a little bit like a different setting, because these parents already had, most of them already did like 4 or 5 different trials, and so they were quite used to going in and out of the hospital and getting all these questionnaires filled out and everything. So I'm not sure if it was really that kind of effect, but I do think it's really important to keep that in mind when analyzing it. Yeah.
Thank you very much.